I'm going to talk about antibiotic resistance, your microbiome and you. And I've scheduled this lecture in such a way that, for example, I think probably the first 20, 25 minutes, it's sort of an overview of the field, first of the microbiome, and then of antibiotic resistance. And then by the end of it, I'm just going to talk a little bit about some of my own research. I've tried to keep the technical bits to a minimum, maybe with the exception of some of the last slides. So I do hope that you will enjoy this lecture on antibiotic resistance, the microbiome, and yourself. I'm going to talk about the microbiome first. So here on the left, you see us, so Homo sapiens. Our bodies consist of roughly 3 times 10 to the power of 13 cells, and people working on human genomes, researchers working on human genomes tell me there's roughly 30,000 genes in our genome. And as a species, as humans, we're actually very clonal. Of course, there are major differences between individuals, but on a genetic level, there's actually not that much difference between us. There's not people on the planet that have a genome that's 30% different from somebody else. And it's also very hard to change your own genome. With CRISPR-Cas, maybe we can start doing it, but it's still very hard. So not only do we have our own genes in our body, but we also have our microbiome. So actually, that means that's the term for the bacterial, mostly bacterial ecosystem that is living on our body. And actually, there are slightly more bacterial cells on and in our body than there are human cells. So for bacteria, we are a great sort of walking incubator, constantly at 37 degrees and always providing them with food. And not only do we have many of these bacteria on our body, they also have a vast amount of biological diversity. So our microbiome is estimated to have more than 2.5 million genes. And the interesting thing is that our microbiome from one individual to another is very different. So, and we can also start changing that microbiome if we want. So that's one of the reasons why this microbiome has become such a field of intensive research over the last decade or so. So where are these microbes? Well, essentially they're everywhere on our body that is in direct contact with the outside world. And if you just think of your body as basically a cylinder, there's a tube running through it, which we call the intestinal tract, and basically that also means that is in direct contact with the outside world, and that's actually where we find the highest number of microbes. So in our gut, we can find trillions and trillions of microbes. So as a start on this microbiome, I'm actually going to give you a little bit of a historical perspective, and I'm going to start with the father of medicine, Hippocrates of Kos, who lived more than, more than 2,000 years ago. And he stated that apparently all diseases begin in the gut. And this is still sometimes wheeled out as a reason why people should be studying the microbiome, the gut microbiome. I've scanned all his writings, and maybe I've missed something, but I actually think he didn't write this. This is some kind of urban legend that is sometimes being told to people that basically already the ancient Greeks thought that the gut microbiome was very important. However, the first person who basically realized that there are bacteria and microorganisms living on our body was a Dutchman, a very famous Dutchman, probably the most famous microbiologist of all time, Antony van Leeuwenhoek. Spent too much of my time explaining to my college how to pronounce that name, but they all know him, so that's good. And of course, Antony van Leeuwenhoek was a brilliant scientist in his own very peculiar way. So if you ever have the time, I do strongly advise you to read, because they're all publicly available, his long and rambling letters that he sent to the Royal Society, where he, for example, in this one, he identified that bacteria were living between the teeth of people that never brushed their teeth. So he was very much the first to realize basically there's life there in our bodies. And then if I now jump forward another 250 years, we end up in the early 20th century, and another great scientist was Ilya Mechnikov. And he is, I'm pretty sure Eva knows him because he's probably the father of immunology. He discovered phagocytes and their role in innate immunity. He also had a lot of opinions about microbiology that haven't really stood the test of time. So he thought, for example, that the microbes that are present in our guts produce all kinds of toxins that would be negatively impacting our health. And indeed, he even foresaw that there might be a courageous surgeon at one point would be removing the colon, which is where we know most of our microbes in our body are, because basically, why do you need a colon anyway if they're full of bacteria that produce all kinds of toxins? I wish it would, everybody, the scientific community would have left it at that, but unfortunately there was a courageous surgeon, very famous surgeon in his time. I think he was practicing in London, Sir William Arbuthnot Lane, who basically did this. He removed the colon because he felt it was unnecessary, and even claimed that sort of streamlining the human drainage scheme was the most satisfactory result of surgery known to us at the present time. Fortunately, over the last century, we've moved forward a little bit, and we now know that our gut bacteria contribute in many positive ways to our health. They help us digest our food, particularly when we eat lots of plant fibers. They produce vitamins. They sort of form a protective barrier against invading pathogens. I think other bacteria like Salmonella that can cause foodborne infections. If you have lots of bacteria in your gut, basically Salmonella can enter the gut but can never gain a foothold because it's completely covered by harmless bacteria that hopefully make up most of your gut microbiome. And also they produce lots of short-chain fatty acids, lactate, butyrate, acetate, propionate, and these are all very important compounds to maintain intestinal integrity. So how do we study the gut microbiome? So as microbiologists, what we really like to do is we like to do culturing. So we take our petri dishes and we try to quantify and identify the bacteria that we grow on them. But for decades, people already realized that this is very difficult. And there's lots of reasons for this. So for one of them is that the bacteria that live on our gut are very sensitive to oxygen. So that means that basically the moment that you have a stool sample and you want to work with that, you have to immediately transfer it to a cabinet that is free of oxygen. And then those bacteria will survive, but otherwise they will die very quickly. And just generally speaking, whereas for pathogens, we sort of know how to grow them on these media that you see here on the left, we don't really know how to do that for many of these, of the harmless commensal bacteria that we find in the gut. We simply cannot grow them very easily in the laboratory. So that's why since the late 1980s, DNA-based methodologies were used to characterize the gut microbiome. And of course, all these bacteria and viruses and fungi that can be present in the gut all have DNA. So basically, when we study DNA, we will be able to capture the entire diversity of all these bacteria. So even though this started in the 1980s, this field of research really took off in the around 15 years ago, when the cost of DNA sequencing dropped tremendously. I know some of you do a lot of genomics as well, so we know that 20 years ago it was incredibly expensive to do genome sequencing, and since around the year 2007 we see this sort of 1,000, 10,000, 100,000-fold decrease in the cost of sequencing. So it became possible to sequence basically all the bacteria that are present in the gut. So once researchers were able to do that, they basically started mapping the microbial diversity of the human body. And basically they figured out that not only do we have lots of microbes in our body, like that astronomically high number of 4 × 10⁻13 that I mentioned before, there's also many differences which type of microbes we find in our body. And they basically reflect a local ecosystem that you can find yourself in. So if you look at this figure here on the left, you see all these sort of pie chart. So the different colors mean essentially different types of bacteria. You can, for example, see that the bacteria that are present on hair, well, if you have it, are different from the bacteria that you can find in the tongue. You can find lots of these yellow type of bacteria there. And again, they are very different from the bacteria that are present in the gut, which you can see here, which are characterized by this dark blue group of bacteria. And that's basically because on hair, you find there's lots of exposure to oxygen, while in the gut, there's hardly any oxygen. pH is very different. So there's all kinds of these reasons to determine which bacteria grow where on your body. So, like I said before, there's many bacteria there, 10 to the power 10 per gram. The composition of the microbiome is almost unique to an individual. There is some overlap when you have identical twins that grow up in the same household. So there seems to be some genetic element to it, but it's also being determined by many other factors, including diet, probably most importantly, but also where you live, whether you've been using antibiotics recently, and lots of other factors that we still don't really understand. So then the question becomes, okay, what now? We know that all these bacteria are there, so what do they do to human health? And so what we know is that the composition of the microbiome has been linked to many different conditions. And so this is just a snapshot of something that I recently found on when looking through the literature. So I'm not going to link them, read them out to you, but it includes obesity, arthritis, cancer, and even sexual preferences, even though that's only been shown in fruit flies to the best of my knowledge. However, we still really don't understand in many of these cases if there's a causative link between the bacteria and the illness. Because often these studies basically compare the microbiome of patients and healthy controls. And for example, patients with colon cancer versus healthy controls. But it's perfectly possible that a patient with colon cancer will have a different microbiome because the local microenvironment in the gut has changed for that individual patient. So, so this is just a sort of caveat that whenever you read something in the news like microbiome has been linked to condition X, there's certain papers in this country, but also in the Netherlands, that almost seem to be publishing these every week. You have to always remember, okay, probably they compared patients versus healthy controls. That's correlation, doesn't imply causation. There's many confounding variables. Nevertheless, this is clearly a topic of intensive research, and don't have the time to really go into that today, but these causative links between different bacteria in the gut and health have been validated in a number of cases in humans. And you can think of, for example, of inflammatory bowel diseases like Crohn's disease or ulcerative colitis, but also there's quite strong evidence now that certain types of cancer also certainly are influenced by the presence of different microbes. Right, I'm going to continue now. I'm going to change gears a little bit and talk, instead of about the microbiome, I'm going to talk about antibiotic resistance, because that's the other major topic of my research in Birmingham. So you all know antibiotics, and you all know that they're crucially important drugs for the treatment of bacterial infections. What many people do not realize, however, is that most antibiotics are natural products. So they are basically, they're produced by bacteria and fungi, and we've basically isolated these bacteria and fungi and made them overproduce these compounds, and then we put them in a pill and give them to patients. So the reason why microbes want to have evolved the ability to produce antibiotics is because in a natural environment, bacteria are essentially competing for nutrients that are very scarce. So you can see a very schematic overview here. The green and the red bacterium both compete for sugars and amino acids. And so the green bacterium can basically win this competition by producing a compound that kills off the red bacterium. That compound, that's what we call an antibiotic, because then the red bacterium will die and the green bacterium can basically metabolize the sugar and the amino acid. And just to illustrate how important these natural products are, is this very famous example of the isolation, or the discovery of penicillin by Sir Alexander Fleming in the late 1920s. So Alexander Fleming was obviously a brilliant microbiologist, but he was also a bit messy. So basically, his lab bench was always a bit of a mess. And in I think it was 1929, he basically shoved all his Petri dishes to one side of his bench and then went on a holiday for 3 weeks. And then when he came back, he checked those Petri dishes and he saw that at that time, a fungus had fallen onto one of his plates. And you can see that here. This is a photo of the original plate. And he could see that around that fungus, this bacterium, Staphylococcus aureus, very well-known pathogenic bacterium, was being killed by the fungus. And instead of throwing this plate away, he basically went, okay, that's interesting. I need to follow up on that. And so in essence, what he had discovered then was penicillin. And of course, that took sorry, I'm stopping here a little bit because I can hear my wife coming home, so I don't have to interrupt this lecture for Sainsbury's later, so she can handle that. So basically, this is the discovery of penicillin as an antibiotic, so a natural product. And so why this is important will come back later in my lecture. And I do hope this is going to work, but just to illustrate how incredibly sorry, I think.
E. coli growing here, and it's living. You see it start to grow, and we're going to have penicillin. What are you going to see? The bacteria.
Okay, sorry. Let me just stop this. So I'm going to show you a video, and I'm not sure whether you can hear the sound. I'll just shut up for a little bit, and I hope you can hear the sound of the video. What you can see is basically bacterial cells growing, and then they basically are exposed to antibiotics, and you can essentially see them explode because of the action of the antibiotic.
E. coli growing here, and it's living. You see it start to grow, and we're going to have penicillin. What are you going to see? The bacteria. They're going to pop.
So you can see the bacteria growing here. Okay, and then they start to explode.
Poke the holes in the cell wall. Boom, bacteria is dead.
Okay, I'm not sure whether this works, but clearly what you can see, this is just showing how incredibly powerful antibiotics are in.
And we're going to go back to the next slide.
in killing bacterial cells. So true miracle drugs. And remember that for most of the drugs that we use in hospitals, you have to take for decades. For example, bacteria against drugs against high blood pressure. You need to use for decades. With antibiotics, you can take for maybe a week, and then you are cured of an infection that's potentially life-threatening. And even though they're miracle drugs, and this is something that is, of course, a concern, they're incredibly cheap. And for the price of a coffee at Costa, you can basically pay for a day of antibiotics in hospital. And for that reason, because they're so cheap, basically they're used very widely, not just in human medicine, but also in veterinary medicine and in agriculture and aquaculture, so that's fish farming. And that has led to this problem, the rise of antibiotic resistance, when these life-saving drugs no longer work. And this has become a real problem globally. So this is some data on the current disease burden. We estimate that maybe 30,000 people every year die in Europe and the United States because of these infections. Compared to COVID-19, that may not seem like a very large number, but the estimates are that maybe by the year 2050, if we don't do anything about this, maybe 10 million people per year might die of these infection. So that's more deaths caused by drug-resistant infections than by cancer. And actually, the problem of antibiotic resistance is far more complicated. It's far more serious in low and middle-income countries, even though the death toll because of these infections has not been quantified in many ways, and because it's very difficult to do that for a variety of ways. But this is just a study from India, where they estimated roughly 60,000 newborns die because of resistant infection, so antibiotic resistant infection. So clearly this is a major concern. And not only that, so the problem with antibiotic resistance is much more complicated as well, because it could really spell the end of modern medicine. And so, as the previous CMO, Chief Medical Officer, said, Dame Sally Davis, and without the use of antibiotics, we can no longer fight infections. And then many of the medical interventions that we now see as routine, like cesarean sections or cancer treatments or hip replacement, where of course an infection can happen as a complication, might become incredibly risky. So that's of course the future that we need to prevent from happening. So basically, I've been talking about, let's say, the microbiome and antibiotic resistance now for about 20 minutes, and I just now basically will try to combine those two. So we know that there's this gut microbiome, these trillions of bacteria in our body, and basically now I'm going to try and address the question, are they resistant to antibiotics? And if so, does it even matter? And I'm actually going to start with that last bit. Does it matter if you have antibiotic resistant bacteria in your body? So here on the left, you can see a sort of general overview of the major sort of groups of bacteria that you can find in an average healthy human's microbiome. Microbiologists love to talk about these type of things. I won't discuss all these groups and all these species in detail, but the most important thing to realize here is that the vast majority of these bacteria are what we call commensal bacteria. They hardly ever cause infections, certainly not in healthy individuals. But roughly 5% of the bacteria that you can find, or 5% or less of bacteria that you can find in your gut microbiome can cause infections, particularly in immunocompromised patients. So the first question that I want to address in this talk where we've done some research on is which of these groups actually carry antibiotic resistance genes. So I'm now going to get a little bit technical. I try to do this as quickly as possible, so to not lose half of the audience. But what we want to do is we want to basically identify which gene is in which bacterium in your microbiome. And we use a technique where we cross-link our DNA within a microbiome by incubating it with formaldehyde. And then with some tricks, we can sequence those cross-links, and then we can identify which resistance genes are in which microbe. And so very happy to discuss this further if you're interested. But basically, this is one of the results that we found, where we link different resistance genes here on the bottom of the slide with different bacteria that are present in a microbiome of a healthy human. And again, I'm not going to discuss all these bacteria individually, but what we show here, and this is just one individual, but we show it in more people, in healthy humans, these gut commensals, not necessarily the pathogens, are major reservoirs of antibiotic resistance genes. You might be very surprised by that. Why would these be resistant to antibiotics, these harmless bacteria? Well, that's a consequence of billions of years of microbial evolution. Microbes have been exposed to antibiotics long before we started using them in the clinic or in agriculture. And that's why many microbes, including these commensals, will have antibiotic resistance genes. But then, does that matter? Does it matter when you have microbes that are resistant to antibiotic, but are unlikely to cause disease? Does that matter? And we think it does. And maybe to highlight that, I'm just going to show you a study that we completed some time ago, which we performed in Bangladesh. So this was a collaboration with the group of Dr. Sirajul Islam of the International Center for Diarrheal Disease Research, Bangladesh, where we wanted to see, basically, where we wanted to sample the environment in Bangladesh, so particularly surface water, and see how much antibiotic resistance do we find in it. And so there's many reasons why we wanted to do this. So we know that there is a major problem with multi-drug resistant infections in Bangladesh, like in India and like in many other low middle income countries. And there's many reasons for this. One of them is that antibiotics can be basically bought in an unregulated way. So you don't have to go to your GP, but you can just buy it at a pharmacy without a prescription. They're used very widely in agriculture and aquaculture, and there's a lack of sewage infrastructure in many of these countries. So to better understand what's going on, basically we did this study to understand, okay, what do we find antibiotic resistance in the environment in Bangladesh? And so these are some photos that I took during our sampling. So basically, and I need to say this for this audience, because Bangladesh is actually just like the Netherlands. It's almost entirely flat. It's at sea level, and there's lots of water. So it's, for me, it was almost like finding a second home away from home. So it was an amazing experience, and we worked there with great people. Very much enjoyed it. But basically, this is what we did. So we had these rural sites in Bangladesh here, which are often used for fish farming. And they have these large ponds in which they farm their fish, and sometimes they use antibiotics to treat or prevent infections in these fish. But also we sampled in Dhaka, which is of course the capital of Bangladesh and one of the largest cities on the planet with 21 million residents. And we used sort of traditional microbiology, where we grew bacteria that were resistant to antibiotics, and we also did some DNA-based methods to determine how much antibiotic resistance we could find. So this is the overview of what we found when we use our traditional methods. And you can see here on the left that you don't see any growth on one of these Petri dishes. That means there were hardly any bacteria there that were resistant to antibiotics. But here in this site in Dhaka, you can see that basically our plate was completely full. So there's lots of bacteria in this site, and this makes perfect sense. Maybe you can see this in this photo here at the back. You can see that we're sampling a river that's next to the slums of Dhaka, and this water is being used to prepare food, to wash clothes. People bathe in it, so it's very heavily used. And of course, it is a major problem when you see so much antibiotic resistance in it. Then we not only did that sort of traditional thing of quantifying bacteria that were present in these sites by growing them in Petri dishes, but we also did a DNA-based survey of antibiotic resistance. And I'm going to skip this slide very quickly, but the point here is that we find many antibiotic resistance genes, and we find particularly high levels of antibiotic resistance genes in these urban water samples that we identified in Bangladesh. And this is something that we were very excited by, and that in some way, we didn't find that surprising, but it was very great to see, is that there was a very strong and highly significant correlation between the abundance of human gut bacteria in these sites and levels of antibiotic resistance genes. So basically, that means that if bacteria that come from the human gut enter natural ecosystems, they bring with them all their antibiotic resistance genes. And I didn't really have the time to explain that today, but these antibiotic resistance genes can spread from one bacterium to another very quickly. So of course, this is a major problem, because this means that these antibiotic resistance genes that are present in these natural environments can basically spread very quickly to a wide variety of bacteria that are present in these ecosystems. And because this water is being used for all kinds of purposes, there's a very high risk of them being return back to humans and then causing infections in people that, for example, are elderly or immunocompromised, and then or very young, and then causing infections that are very difficult to treat. So to conclude, we find that very strong correlation between the abundance of human gut bacteria and antibiotic resistance genes. The human gut is a very important reservoir of resistant bacteria. And as a sort of a policy point here is when people talk about antibiotic resistance, one of the things is always they say, okay, we need to have new drugs. And yes, that is very important. We need new antibiotics to treat infections caused by antibiotic resistant bacteria. But we might also want to think about whether we want to better invest in sanitation and sewage treatment as interventions to reduce the spread of antibiotic resistance in low and middle income countries. Because just like with COVID-19, these bacteria travel. So they basically come to us at some point, and we can see in a city like Birmingham, where we have lots of people traveling to and from the Indian subcontinent, we do generally see higher levels of antibiotic-resistant bacteria than in rural parts of Britain, where maybe there's not so much travel with international travel occurring. So we need to think globally to solve this problem, and I think one of the best ways to do this is to prevent the further spread of these bacteria rather than trying to treat them with antibiotics, new antibiotics that bacteria will become resistant to at some point as well. So my future work is basically focusing now to better understand antibiotic resistance in the gut microbiome of critically ill patients. We already know that basically in these patients, the microbiome is changed completely, and it's actually overgrown with some of these opportunistic pathogens that can cause infections. So we want to better quantify whether that's a direct threat to their health. And indeed, we want to study interventions to reduce the carriage of antibiotic-resistant bacteria in the gut of patients to minimize that risk of infections. So I want to leave it at that. I want to thank the people that I work with at the ICDDRB in Bangladesh, my funders that funded this research, and my group in Birmingham, particularly Greg McCallum, who did that first bit about the identifying bacterial hosts of antibiotic resistance genes in the gut, and Ross McInnes, who led that study that we did in Bangladesh. And I would like to thank you for taking the time to listen to this lecture, and for Ewout for inviting me. So thank you very much. I hope there's still some time for questions.